Huge aloha from the Big Island of Hawaii. We are a team of faculty, staff, and undergraduate students from the University of Hawaii at Hilo. Our university is located at the east side of Hawaii Island. We offer a variety of undergraduate programs along with select graduate programs such as pharmacy, indigenous language revitalization, and tropical conservation biology. The work that we're presenting today is a result of a collaborative effort between the computer science, marine science, and the art department. This work is supported by the Academy for Creative Media System. My name is Francis Cristobal. Again, welcome and thank you for joining us. I am the faculty specialist in the computer science department, and I am the project lead of the Coral Reef VR software. I'm also the technical lead for the Academy of Creative Media System at Hilo, and my research interests are in data visualization and emerging technologies. Dr. John Burns, unfortunately, will not be able to join us today due to prior engagement, but he is an associate professor at the Marine Science Department at Hilo, and also the director of MegaLab. His research focuses on the coral reef and community ecology. Dr. Burns uses innovative technologies to enhance our ability to study and conserve this valuable ecosystems.
I'm Michael Dodge, an undergraduate engineering student at the University of Hawaii at Manoa. I served as the VR project's lead developer in its early stages.
Hi, my name is Kailey Pascoe, and I'm a coral reef research technician for the MegaLab. And my involvement with this project was organizing the 3D models and informational slides for each site.
And my name is Alex Spengler. I'm a recent marine science and data science graduate. Together with Kailey, I work as a research technician for the MegaLab, and I'll be your tour guide today.
All right. Well, the conservation and preservation of coral reefs in the Hawaiian island chain remains one of the critical missions of the marine scientists of today. A wide variety of tools, visual tools and techniques are utilized to support this mission, but are typically bound to a desktop environment. So this project uses a virtual reality technique to visualize several coral reefs found throughout the Hawaiian archipelago. The core of the application involves recasting 3D data collected from the field into a fully immersive virtual reality environment. A custom user interface is used then to support navigation and manipulation of this data sets. The 3D content used for the application was derived from a set of photogrammetry models constructed from high-resolution scans of the Pacific coral reefs. The data collection originated with an annual research expeditions and collaborations with NOAA's Marine National Monument located in the north of Hawaii to improve the understanding of how habitat structures affects associated reef organisms and large-scale ecological processes. Geospatial analysis was applied to the models in order to quantify and classify characteristics of this underwater habitats. And I'm going to send you to Kailey to talk about the process of doing the photogrammetry system.
Thanks, Francis. So coral reef ecologists have long recognized that 3D structural features of coral reefs influence important ecological processes. However, due to the high cost and difficulty of applying 3D techniques into your average monitoring program, coral ecologists have only recently developed practical approaches for quantifying 3D characteristics of coral reefs in the last 5 or 6 years or so. These new approaches uses structure from motion, SFM, photogrammetry, which is a cost-effective automated range imaging technique for estimating 3D structures from 2D image sequences. SFM photogrammetry uses overlapping imagery, with a scale invariant feature transform, SIFT, which is a feature detection algorithm that detects and describes local features in an image. And we can often see this on land being done by drones or aerial imagery, but when we take this underwater with us, we become the drone swimming over the coral reef structure, taking images. So today, the sites that you will be touring are long-term monitoring sites in the Hawaiian archipelago and the Papahanaumokuakea Marine National Monument. And they are surveyed at least once a year using SFM photogrammetry. Scientific divers use scuba to collect the overlapping imagery with a single lens camera. And we can collect anywhere from 200 photos to 2,000 photos, just depending on the area size that we're surveying, in which we take those images later and render them into a 3D model using a software called Adasoft PhotoScan. And the 3D derived products, such as point clouds, digital elevation models, and orthophotomosaics, have high precision and accuracy across multiple spatial scales. And this is huge because we can bring the reef to the lab, and the ability to create such high resolution 3D models of coral reef habitats and dramatically enhances our ability of research. And in addition, there are great visuals for outreach and education of the Hawaiian coral reefs, because many of these sites you'll be seeing today are very remote and hard to access. So now Michael will be speaking about the integration of our 3D models into virtual reality.
Thank you, Kailey. So at the start of the project, Dr. Burns selected 8 of these 3D models from Mega to be featured in the project. Now these models took a little bit of extra preparation to bring into an interactive VR environment. I applied some simple mesh editing techniques in Blender to bring the polygon counts down by about 80%, small enough that the engine can handle them without requiring any special middleware or code. Once the meshes were prepared, I imported them into Unreal Engine and implemented them as a generic actor object with some extra code to allow for future scripting. Each of these reefs were implemented as independent levels to keep the workflow relatively simple and to keep loading times on the order of just a few seconds. The main structure of the museum was created in Autodesk Inventor and textured using freely available designs. To visualize all of this, Alex will now give you a walkthrough of the project.
Thank you, Michael.
One of our primary objectives in the Mega Lab is to take innovative technologies and apply them to our research so that we can improve our understanding of both the biology and the ecology of coral reef ecosystems. We create these high-resolution 3D maps, and then we can overlay as many forms of data as possible in order to discover how these reefs will respond to large-scale stressors such as climate change and local stressors such as pollution. In addition to this cutting-edge work, we're able to virtually preserve these sites and use virtual reality apps such as this one to actually take you to these research sites where you would never otherwise be able to explore. We hope you enjoy.
All right, so welcome to the Hawaiian Coral Reef Museum by the Hilo Mega Lab. I'm Alex Spengler, and I'll be your tour guide today. To navigate through these different environments, I'm primarily using the Oculus Rift hand controllers. The analog sticks move me in the 3D space. I use the left menu button to hop in or hop out of the 3D models, and the trigger to grab the little globes at each of the different models. So I'm gonna head over to the right here and kind of begin the tour at a really unique reef on the Big Island of Hawaii, the east side of Hawaii, known as Waiaupai. Alright, so I'm right here, kind of in front of the plot, and to navigate through the plot, I can use the analogs on the controllers, the analog sticks, to kind of move and almost swim through the 3D space, or the right trigger to kind of navigate up and down, and I can just point and swim and navigate through the plot. There's a headlamp located in front of me. Let's see if I can get closer, and you can kind of get a idea of where the headlamp is right here, almost as if I was swimming around in an actual reef. I might have a light with me, and you can kind of see that illuminating the different areas around me. Another feature that's built into the VR system here is scaling of the plots. So I can use the left trigger and use the left trigger to actually scale and kind of shrink the plot. And it's going to be more of like almost as if the reef was like the size of a tabletop. Or I can expand it to get different areas of scale. Oh, here we go. Now I'm doing it. So I'm shrinking it down here. You can see that the reef is a lot smaller. Or I can expand it. And I, in turn, are going to be kind of smaller in the reef. Right. And another interesting feature that's built into this VR app is this Explorer mode. So you can see the notification in front of my pointers here. It says press B to toggle exploration mode. So if I press B, these little icons appear throughout the different plot. You can see one there and one a little bit farther away. And if I swim over, they'll give me some information about the reef if I press A, or little points of interest. So this sign is, or this little placard is describing Waiaupai, and I chose to start off at Waiaupai because it's a particularly unique site. Interesting term that Dr. John Byrne used when he was kind of giving that intro video was virtual preservation. In 2018, this site and all of Waiaupai was covered in lava. So the recent volcanic activity in 2019 encased this whole reef under lava and actually extended the shoreline about 3,000 feet further into the Pacific Ocean. So this is an area of reef that's literally frozen in time, and it provides a whole lot of unique information for future researchers can use existing plots and models to run different statistics on, or it can be used as an outreach tool. It's kind of like a historical example, a true museum. You can see something that doesn't exist on planet Earth anymore. We can ahead and take a look at a few more of the different placards, or the different info cards, and this one's describing some coral species, Montipora capitata, or a rice coral. They almost look a little bit like lichen, where they encrust, like what my cursors are pointing at over here, encrust and lay pretty close to the ground. And they're called rice coral because they have these little varicae, these little bumps throughout their whole surface that look a little bit like granules of rice. This is describing a different species of coral, Priades lobata, and they're lobing coral or mounding corals. Throughout the plot here, and some other plots we'll look at, they're characterized by this kind of yellow or brown coloration. You can see a whole bunch around me here in the reef. And as Kailey mentioned in her overview, when the Mega Lab or researchers enter these reefs and actually collect the information or the photos that we use to make these models, we use a DSLR camera, a single lens camera, encased in an underwater housing, and typically begin on one side of the reef in a specific area, and kind of like my hands here, swim over the reef and click, 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 take a whole bunch of different photos, and eventually we get to the other side. I'm not a very fast swimmer, but if we turn around here, we can slowly take some more photos, and we want to ensure that there's enough overlap between those two tracks because we use a program called Metashape that stitches all those individual images together and actually builds a 3D model. So if we have enough overlap between sequential images, the software uses a SIFT algorithm and gets some stereo information from nearby images based on the elevation of the camera and the plot. And to get a sense of the scale of what I'm looking at here, you're going to see these in various plots, but our lab uses ground control points, or GCPs, to scale the individual photographs. So that allows us to prevent things like image distortion from lenses or whatnot. So we usually put these ground control points, that's kind of what I'm facing or outlining here. The ground control point from this red marker to the corner here of the ground control point is about 10 inches or about 25 centimeters. So this is about a 25 centimeter in diameter lobing coral here, Montipora capitata, or I'm sorry, lobata. We're gonna back out to the museum and take a look at some other 3D sites here located in the museum. So over here on the left side of the museum, we have a site in the northwestern Hawaiian Islands. Now the northwestern Hawaiian Islands extend from, if you were to kind of superimpose them over the contiguous US, would actually extend from Oregon all the way down to Mississippi. So they're quite expansive. Another interesting thing that you might see kind of swimming around me here in the reef, or with me here in the reef, are these triggerfish. This triggerfish is the reef triggerfish, the state fish of Hawaii. You can see that they're kind of 8-bit reef triggerfish. Now this island, Lisiansky Island in northwestern Hawaiian Island, is characterized by a different species of coral than we saw in the previous plot. The corals here have a whole different hue. They're kind of purple or blue, and that's because this reef is characterized by purple rice coral. Now, that purple coloration comes from a endosymbiont, or a symbiodinium, a microalgae that actually lives inside of the coral animal and allows the coral to get some photosynthetic energy. And usually when people are talking about corals, at least in a modern context, the term that comes up quite a bit is coral bleaching. We can actually see the beginning stages of coral bleaching over here, and that's kind of what makes this plot unique in the VR museum that we have, is this survey was taken in 2014, and frozen in time is actually the beginning stages of coral bleaching. So you can see some white corals over here that are a little bit more pale, and in fact this information placard or card describes the coral bleaching process. And I could talk a little bit on this. It's similar to like a fever. The coral bleaching is a stress response, or a response to some stress in the coral animal, whether that be a global stressor like increased ocean temperatures, or a local stressor like pollution runoff. The coral will actually eject or spit out that endosymbiont, that microalgae, and that leaves the coral kind of looking pale or almost translucent. It will lose that purple or brown or yellow, whatever color it was before. It'll lose that coloration and kind of be more pale. And that leaves the coral more vulnerable to being overtaken by different opportunistic organisms. So I'm going to back out using that left button on the controller there. You can see my hand. And we're going to visit the same area, also on Lisiansky Islands. But as the information placard here says, this scan is in 2017. So this is 3 years after the other scan was taken. And we can see kind of how things have changed after that bleaching event in 2014. So here's the same area of reef in the northwestern Hawaiian Islands in Lisiansky Islands. And you can see that things look quite a bit different. There's a visual difference. There's no more purples or blues. It's kind of, at least in a color palette, characterized by more pastel colors, browns and yellows and tans. And there's something kind of interesting happening on the far end of this plot. You can open Explorer mode again and take a look at this placard, and it's going to tell me what's going on here. And that's a coral algal phase shift. So the coral algal phase shift happens after the coral is vulnerable or the coral dies. The macroalgae, all this kind of green stuff over here, will slowly overtake that vulnerable coral. It's fighting for the same resources in the marine environment. And one of the parameters or factors that the Mega Lab analyzes is things like structural complexity or rugosity. Basically, how many nooks and crannies, little caves, are located in a marine environment. And this is an oversimplification, but typically the more nooks and crannies, the more complex an environment is, the more places there are for fish and invertebrates to hide. And typically a more structurally complex environment will allow a greater abundance of biodiversity and more animals can live there. So the algae will come in like this cactus algae or Halimeda and overtake the environment or the area in which the coral used to inhabit. And the algae will actually break down that calcium carbonate skeleton and kind of smooth things over, leaving behind a less structurally complex environment. You can see another GCP, like I mentioned earlier, one of those tools that our lab uses to scale the models. We can swim up and take a look and then back out to the museum here. So I'm going to head to the other side and look at a different kind of plot. So this is located on Morrow Reef. And that little sign in the museum might have led you to realize that this isn't exactly a coral reef, but a wreck. This is the UNSN San Miguel. It's a World War II era tanker that actually ran aground at full speed and full ballast in 1957. Now, there were no casualties or fatalities on this wreck. Everyone survived and was able to escape and be rescued. But this was actually discovered in 2015 by a snorkel survey crew that was just snorkeling over the area. Again, I mentioned just how expansive the northwestern Hawaiian Islands are. Again, ranging from, if you were to superimpose them over the contiguous U.S., from Oregon to Mississippi. So it's easy for something even as large as a huge tanker to be lost. To get a sense of the scale here, I can swim down into this ladder here, towards the ladder, and just to get an idea of how big this model is, or this actual ship. Or I could shrink it down using the scaling feature, or the scaling control, and we can have like a little toy ship. But this discovery was surveyed by our lab in 2015, and we were able to produce a paper based on the archaeological importance of this site. So now it's quite a bit smaller. It's like my whole body is the size of that ladder now. All right, I'm gonna back out to the lobby of the museum. We're gonna look at some more plots. Another really interesting plot is this one right next to the UNSN San Miguel. This is a site located in French Frigate Shoals in the northwestern Hawaiian Islands. And it's characterized by a different species of coral than we saw in the previous plots. So if I, again, turn on exploration mode and head to one of the navigation points, or these little info points, I can see that the corals that are surrounding me, these corals that look different than corals in other plots, are table corals, or members of the Cropera genus. These are much more abundant or prevalent in the northwestern Hawaiian Islands than the main Hawaiian Islands. And this site was particularly unique in that it housed a high biodiversity of fish. A lot of beautiful photographs came out of this area just based on the sheer abundance of tropical fishes. Now, when our team went to survey this site in 2018, they went to the GPS location of this exact reef, which our team does year after year, or over sequential years to kind of check on things like change, species dynamics, and rugosity, or those structural complexity dynamics, and see how things are changing from year to year. But in 2018, there was a hurricane that came through the northwestern Hawaiian Islands and actually obliterated this whole reef. So in front of me, you can see that picture in the info card of the shark swimming over that barren, rubbly reef, and that's actually the same location. So this is another great example of that virtual preservation, or an example of those sites that are kind of preserved in time or only exist in this virtual museum now. And we can even use this exact plot and revisit the same area year after year to see how things are growing and look at settlement patterns, because that hurricane and other tropical storms are largely just a normal part of the marine environment, and we can look at how this site is growing and how juvenile corals are settling in this area. You can take a look at this little orange thing on the ground here on the ocean floor. That's a transect tape or transect reel. So when our team visits these areas, they follow that GPS location and lay down transect tape, and that gives them a set distance, and they can survey the same distance and again kind of swim using scuba in that zigzag or boustrophodonic pattern. You can see the tape there laid over some of the corals, as well as GCPs and scale bars over here. I'm going to back out to the museum and look at one more reef. So over to my right here is Kiri Atoll. It's also in the northwestern Hawaiian Islands and is at the northwestern part of the northwestern Hawaiian Islands. And the Hawaiian Islands are created as they travel over a hot spot in the Pacific Plate. And as that Pacific Plate glides over the hot spot, different islands like Kiri Atoll get farther and farther away. This site also looks a little bit different in that it's characterized by yet another species of coral. This is a cauliflower coral, or members of the Pascillopora genus. All these kind of globular structures all around me are those cauliflower coral. And Kiri Atoll is located at a unique place in the northwestern Hawaiian Islands, again being so far from the main Hawaiian Islands that this island is actually beginning the end of its life. It's at a point we like to define as the Darwin point. So this describes a point in Darwin's theory of reef ecology and reef creation, where there's kind of a natural process where the big island is the largest island and one of the youngest islands, and as that plate drifts farther and farther away, there's no hotspot, there's no volcanic activity, and the islands kind of sink back. Corals have that photosymbiotic symbiodinium, that microalgae living inside of the coral animal, and they need sunlight in order to get energy and metabolic function. And as islands like this atoll, Kiri Atoll, get farther and farther away from that hotspot, they sink lower and lower. And Kiri Atoll is at a point where it's so far north that this island is, eventually going to sink below the photic zone, and these corals aren't going to be able to get any photosynthetic energy. I can back out to the menu here, and I want to give a special thanks to Dr. John Burns and Francis and Jason and Christopher, as well as many other members that help make this virtual museum possible. And that's it for me. Thank you for coming along on my virtual tour.
This will be offered as an open source format, so we are going to be providing this for free download and free use. If you're interested, email us at uhhcs@hawaii.edu. And if you also want to know about more of the Megalab project, you can check out the website at themegalab.org. And you see more of their project, what they do, and be able to look at more of the coral reef models out there. But on behalf of Alex here, Mike, Kailey, and John Burns, and all our team, thank you for joining us, and aloha.